for posting. All right, can you see the slides? Just give me a thumbs up or a, yep, perfect. Okay, so welcome everybody. Thanks for joining today. I guess this week was out of this world. Hopefully that was maybe out of this world good, or hopefully not out of this world bad, but just crazy. But yeah, so thanks for being here. We're gonna jump into population genetics and Mendelian genetics, and it's gonna be super fun. And we're gonna do a little bit of interactive activity. And then this morning or earlier today in the session, we did some of the math equations on a kind of whiteboard screen, and that seemed to really help folks. So I'm gonna go through that as well. So before we move on to talking about the genetic stuff, a few reminders for your post lab that's due tomorrow by the end of the day. Just remember kind of typical graph stuff. So always label the axes on your graphs, and then definitely make sure to title your graphs. So you have the 3 graphs to do, so make sure they are looking tip-top ship shape for turning in. I really recommend watching the how-to videos for making the graphs, for going through the t-tests. There's one made by a former TA that is extremely helpful in going through how to add those standard error bars. And I really recommend it. If you just kind of do it step by step with the video, it makes it a lot easier to do. So that's a great resource. And then I have my office hours tomorrow morning. So you can also come to those at 9 a.m. if you want to go through the stuff or talk about homework question, post-lab, lab materials, etc. So before we move on, does anybody have any specific questions they wanted to talk about, go over in class? Type them in the chat if you want, if you have them. Okay, I'm not seeing anything. There were 2 questions actually on the pre-lab from this week. I know it was a doozy. You are all champions for getting through it. It was many questions, but this is really complicated and also important stuff. So hopefully you learned through doing it as well. On question 12, they were asking about frequencies. And this is just a reminder that a frequency is like the decimal out of 1 that makes up the proportion that you're talking about. So for example, if they're asking about the proportion of flies with, or the frequency of flies with red eyes, that would be the number of flies with red eyes divided by the total number of flies. So some people put in percent, and that was fine, but really they're looking for like the decimal frequency of that. So that was just a reminder for that one. And then for question 18, this one was a little bit trickier. They had that lovely little fly family tree in the lab. I know I'm supposed to love all creatures, and I do love all creatures, but I do find insects a little bit scary. So this full page of like up close insect pictures was a little bit intense, but so don't know if anybody else felt the same way. But yeah, so we have this adorable little insect family tree. And in question 18, they asked if there were any phenotypes that would be impossible to obtain if the presumed father were the only father of this family. And so that was yes. And then the answer was, what were the phenotypes that were impossible? And that was any fly with red eyes. And what's important to think about here is that above you were told that the non-red eyes was a recessive trait. So that means that both alleles for both parents would have to be, both alleles for the individual would have to be the recessive ones for that individual to have the sepia eyes. And so since both parents on this diagram, the female and the male, have sepia eyes, but then fly number 7 down at the bottom has red eyes, we know that there has to be another parent involved, because if both parents, all 4 of their alleles controlling eye color are for sepia, where did that red one come from? So that's the thing to think about. And some people said identified fly number 7 or that it was red and brown, but really we're talking about the red eye phenotype trait. And so a way to tell that on a question would be like that they're talking about what are these phenotypes? So talking about the phenotypes that you couldn't have if you were talking about the, if you were looking at the traits of the flies, in this case, the eye teller. Any questions about that question? Yeah, there were definitely some tricky questions in there, and overall people did really well. So good job. Okay, so let's also feel we can come back to this. So type any questions as they come up as well. All right, so this lab we'll be going through genetics in 2 parts, kind of starting at the individual level and looking at principles of inheritance, so Mendelian genetics, and we're going to be doing this paper chromosome activity, or you can also do an online version for people who are doing it at home or who don't have the lab kits. And then if you want to do this stuff individually, also and I recommend everyone goes through these videos too. There's an introduction to the activity and then kind of going through these little, what these chromosome flaps are, because I looked at them and I was like, what are these at first? So I watched this video and it's very good at explaining it. We'll go through it a little bit too, but it can be hard and I don't have a great screen for you to see. Actually, it's better than it was this morning. So yeah, those videos. And then if you didn't receive a kit, you can also go to this link and it has a kind of online virtual interactive way of doing this chromosome activity that is pretty cool as well. And then for part 2, we're scaling up and looking at population genetics. So how do we measure evolution in a population? There's also an introductory video for that. That's really cool. So we're kind of thinking about genetics on these 2 levels. So there's this individual, what traits and genetics and alleles get passed from parent to offspring, and then large scale, how are populations changing and kind of what's controlling those changes. And then something to note for this lab that's different from the other ones so far is that instead of turning in a document PDF, you will be turning in a Google Forms fill out sheet. So the link is on Canvas under the Lab 5 assignment, and there's the form, and fill it out for your post lab. Okay, so diving straight into Mendelian genetics, kind of more on the individual, smaller scale level. So what's the deal with it? You have kind of gotten introduced to this topic in lecture and through the pre-lab, and probably many times throughout biology talking about Mendel and his little pea plants. But we're going to be going through it hopefully more in depth in this class, and hopefully you'll get an even better understanding. So to start, what is a genotype? Can anybody tell me in the chat or out loud? Feel free to unmute yourself. What is a genotype? You can do it privately also. Yeah, so yeah, genes. This is what genes are made of. Genetic material, yeah. Our DNA, what makes us who we are. Alleles, great. So how many alleles do you need for a genotype, Katie? For the traits that we've been looking at. 2, yeah, great. So this is key here. I think that sometimes kind of the difference between alleles and genotypes can be confusing. So the genotype is the pair that you have, like what the coding for that trait is, and then the alleles are the specific what is making up the genotype. And everything, so what we've looked at is like you'll have the big S, little s, and so an allele is the big S or the little s, and the genotype is the 2 together. So that is kind of the critical, that's the difference between those 2 things. And then what is a phenotype? Yeah, so the expression, the physical traits of the organism, physical characteristics, all great. So in the example here on the slide, say we have this big W, that's the genotype. And then that codes for the phenotype of having the wrinkled wings. So it's how are those alleles that make up the genotype being expressed. So that's the phenotype. Then kind of the other half of understanding this is how does the genotype contribute to the genotype of the parent contribute to the genotype of the offspring. And kind of important processes to remember here is probably something you've also learned about a bunch in bio in high school is, you know, how sexual reproduction occurs, and you have the gametes form and then fertilization and then the genotype of the offspring, meiosis, mitosis, all these fun things. But at least for kind of how that relates to this lab, you're thinking about what is involved in gamete formation. So can anybody tell me why gametes are different from many other cells? What is unique about gametes?
Is it because they carry the genetic makeup, like the genotype?
So so all
DNA.
the cells will carry genetic material, but there's something different about the genetic material in the gamete cells. Yeah, I'm seeing haploid. So totally right that, yeah, they carry the genetic material that is important for making the genotype of the offspring. But what's key here is that they are haploid. So they only have half of the genetic material. And we talked about that you need the two, you need a pair to make up a full genotype, full genetic code to make an individual. So these gametes just carry half of that. And then when fertilization occurs, then they mix up and they find their match, and then they create this genotype of the offspring. And that can be, they can find a match, and it can be this homozygous pair, which means that, say it's the big W and the big W, say that they meet each other after fertilization, then that's homozygous, or it could be big W, little w, and that could be heterozygous. So what's important to remember here is when the gametes are formed and you have just these haploid cells, then they can carry any, you know, portion of the parent's DNA, and then it's kind of this segregation and then random mixing when fertilization occurs. And so the genotype of the offspring is kind of due to some kind of chance in that way, that it's whatever genetic material, half of the genetic material happens to be from both of the parents. So, and you'll learn a lot more about that, I'm sure, through this class and then through the BIS series, but it's pretty crazy. Okay, so we're gonna attempt to do this activity. I didn't have anybody with their videos on earlier because of internet stuff, so it was a little bit harder, but we're gonna try and do it. So does anybody have their little cards? You can tell me if you do or hold it up. I don't see. Nobody has their little cards. Okay, so Young has hers. Do you have yours? Your card?
Yeah.
Okay.
Wait,
awesome.
we're on week 5, right?
Yes, we're on week 5. Okay, great. We're gonna do this. Perfect. All right, and Su Young has hers too. So, okay. So to go through these, basically you have 2 cards. For the folks at home too, you have 2 cards so that you can do this with yourself if you want. But for right now, for doing it live in the lab, you'll only need one of the cards. And I'm just gonna pick one randomly. And then, so what we're gonna do is, Suyoung, you're gonna lift just one of the flaps. So you should have 4 flaps cut out, and you're gonna lift one. Both of you, Smriti, you're gonna do this too. You're gonna lift one flap randomly from the left side, which are the R genes, and then one from the right side, which are the S genes. And let me know when you have done that.
I'm sorry, I didn't actually cut it. So I have to cut it and then.
Okay, yeah, you can also just, yeah, do you have the materials to cut with it?
Yeah.
Okay, thank you so much. You're doing great. This is great. This is what this is all about.
So on the lines, right? Like
On the
here.
line, perfect. Okay, see chat. Okay, we're actively cutting now. No worries. All right, and so basically, as they're cutting, as reproduction is actively happening now, as they're cutting, I'll just go through this. So, and this is, you can read about this also in the lab manual, but you have 2 alleles on the S chromosome, and then there's the R chromosome. And so pretend that these are just 2 chromosomes in this creature, whatever creature you want to pretend that it is. And the H chromosome controls head shape, and the R chromosome, sorry, yeah, not R, F, sorry. The F chromosome controls foot heat. And so we're going to randomly mix, and we're going to make an organism that is some combination of these alleles that creates a genotype that then leads to a phenotype. And this is just to show you that, like, an individual will have both R and S, but when you have the the haploid gametes, they would just have 1 R and 1 S rather than 2 R's and 2 S's. And so now you, by pulling up the flaps, are making 1 haploid gamete, and then when we combine our 2 haploid gametes, you will create a full individual with both alleles for these 2 chromosomes. I know that, hopefully that makes sense. All right, so we have our demoers who have heroically cut their little guys. Can you show us or tell us the colors for the one with the black on the top? So that would be the R chromosome, and the one for the S chromosome. Okay, just do one of each side. Okay, so we have what looks like a green, and is that red or pink? I can't.
I think it's red.
Okay, so we have a green and a red, and then our other gamete is coming. Okay, we have a light blue, and then from the other side?
It's light blue and black.
Yeah, and then one from the other side of the flap, one with the pink or red.
Oh, it's red.
Okay, and a red. Cool. So our individual that we have created now has, for its R chromosome, so the ones with the black tops, we have a light blue and a green. And then for the S chromosome, we have 2 red. So it's going to be homozygous for the red. And so for the 2 red rectangles, it's homozygous. And this is on page 2 of part 1 of lab 5, if you want to look at it. But to look at the homozygous 2 red rectangles, that means that our individual that we have created has a round head. Very cute. And so now then we'll go back down to the foot temperature one, which was the ones with the sorry, the head color is, wow, I've written different things on this than I'm supposed to, but it's okay. So the ones with the red are the head color, the ones with the blue or green or yellow are the foot temperature, and we have a dark blue and a light blue. And so this one, there's partial dominance. I'm sorry, we have light blue and green, my bad. Light blue and green, and if there's at least one green rectangle, you just have to follow along with this, then it means that the foot temperature is hot. These are funny traits that they have selected for us to look at. But does this activity make sense to you yet? I'm sorry, I know it's kind of hard to do in this format. We can go through another one if you want.
There's also white. I'm not sure, like this white.
Yeah. Yeah, so the white and black.
Also,
oh, sorry, go ahead.
Oh, no. Like, I didn't understand, like, what, I'm so confused right now.
Yeah, I totally understand. So the white and the black on the top, like you were showing, is to identify which chromosome we're talking about. So the one with the white on the top is the one that controls the head shape. The one with the black on the top is the one that controls the foot color. So when you're doing this activity, you're gonna pick one from the white block top and one from the black block top. And that is your haploid gamete cell and the chromosomes that are contained in it. And then your partner or yourself, if you're by yourself and doing this, your partner will do the same thing. And then you'll put your two chromosomes together. So you'll have two with the black top and two with the white top. And then you have created one complete individual who has two genes that will make a phenotype.
Oh,
Does that make more sense?
Yeah.
Great,
Okay.
okay.
Table, it really helps.
The table?
Yeah.
Yeah, so definitely for, if you're watching this later, I recommend what I did for trying to figure this activity out also is I watched the video and then I read the lab and then I tried to do it myself. So I really recommend going through it. And if you have a friend in the class, you can do a fun game where you make different individuals and see what kind of foot temperature and what kind of head shape they end up with. So yeah, thank you so much to my brave participants for that. That is, I know it's hard to do that on here, so I appreciate it. Great, and then if you don't have the boxes, you can, the lab boxes with these fun little papers, you can make them yourself, or you can also click the link to do it interactively. If anybody desperately wants us to go through another round of that, we can. Otherwise, we can move on to the next part. You can also chat me privately if you need to do that again. I know it was really fun. Okay, well, we can do it later after the recording also. Okay, we did this. Gamete forming activity, so fun. Okay, so moving on to part 2 of the lab, population genetics. So I just tried to find a creature with a lot of animals all together for the population picture. We're not, unfortunately, doing a population of penguins lab. That would be super fun. No, unfortunately, it is with these little guys, the fruit flies. But I guess they're cute in their own way. All right, so scaling up to talking about population genetics. So is evolution occurring in our fruit fly populations? And if we were in real life, we would be counting the fruit flies ourselves, but we have these counts, but really we're gonna focus on trying to understand the math and this predictive Hardy-Weinberg population dynamics situation. So how are the genotypes that we're observing and the allele frequencies How is that changing? How are the observed numbers different from the expected numbers? And what does that say about, is there evolution happening in our populations? So I'm gonna walk through, I'm just gonna explain through this. It's a lot. So I'm gonna go through it once and then I'm gonna go through it again kind of on the whiteboard thing. So if you feel like you totally got it after I walk through it, through the slides, you can feel free to pop out after that. But otherwise, I'm going to do more in-depth, like walking through each step. And that seemed to be really helpful earlier. So we'll do that too. So if it's kind of confusing this first time, don't worry, we'll go through it again. So this is this experiment where you have this initial population of fruit flies. There's like a bajillion fruit flies in this container, and this is called the S0 generation. Then you take one mated female, so all in her lonesome, pregnant, single mother, out to her own little container over here. And she gives birth to babies. And then they end up in this F1, F1 generation. So this is from that larger population. You have one female pulled out who was already mated, and then these are her offspring, or her offspring's offspring, but they come from her. And then you have just the next generation from that big population that all ends up in the same, they're just mating and reproducing, and then there's an S1 generation that comes from that S0. So this is from the large population, this is from the small single female who was taken out. In these populations, the trait that we're looking at is eye color. That happens to be a co-dominant trait, which means that if you're homozygous for this one allele C1, then you have red eyes. If you're homozygous for this C2 allele, then you have white eyes. And if you have one of each, then you have pink eyes, so between the two. So we have two alleles. We have C1 for the red eyes and C2 for the white eyes. And then so we have red, white, and pink eyes, depending on if you're homozygous or heterozygous. So there's going to be a few steps to determining if this population is meeting Hardy-Weinberg equilibrium, meeting the criteria necessary for Hardy-Weinberg equilibrium, or if it's diverging from what we'd expect under that equilibrium. So again, the different genotypes that we have and the phenotypes that they equate with, C1 equals red eyes, C1 equals pink eyes, and C2 equals white eyes. So to begin, step one, what you're going to do is look at the genotype frequencies for the initial population, that big giant tank of flies that we started with. This is like what we did in the pre-lab, where all you're doing to find a frequency is looking at the numbers that you observed over the total number of fruit flies. So we had 214 flies total at the beginning of this experiment, and we had 85 red-eyed flies, 29 pink-eyed flies, and 100 white-eyed flies. So then you just divide each of those numbers by the total population to get the frequencies. And hint, hint, this should add up to 1. So if you add up those numbers and they don't add up to 1, then there's a problem somewhere, because they're just kind of, think of it as like percentages of the whole, but just, with the decimal there. Then after you've found the genotype frequency, so this is basically like, 39.7% of our flies were red-eyed, 13.6% were pink-eyed, and 46.7% were white-eyed. Now, what are the allele frequencies? So remember, we talked a little bit at the beginning about the difference between genotypes and alleles. And here, there are 3 genotypes and only 2 alleles, C1 and C2. So to figure out the allele frequencies, so how common each of those alleles, C1 or C2, is in the population, you're going to take this whole C1 frequency, so 0.397 plus half of the heterozygote one, so half of 13.6. So this is 0.397 plus half of 0.136 is this 0.465. And then you'll do the same thing for the C2. Then you end up with these two numbers that should also add up to 1 as a hint. And this is how common in the population, just looking at all the flies, and you were to count each of the C1 genes, C1 alleles from all of them, and each of the C2 alleles from all of them, what percentage of that would be C1 versus C2. All right, now that we have these allele frequencies, we can move on to step 3, which is calculating the expected frequencies, genotype frequencies for S1. So this would be, we have that big tank, and then we get the original numbers. We come back a few weeks later, and then we see what do we expect to have happened? What under Hardy-Weinberg equilibrium, which has these certain set of rules and expectations, what do we think the fly population is going to look like? And to do this, there's this handy dandy little square that kind of should remind you of a Punnett square. It's same kind of idea, but on one side, you're gonna do the C1 and C2 frequencies, and on the other side, you're gonna do the C1 and C2 frequencies, and you're just gonna multiply each of them out. So C1 times C1, C1 times C2, C2 times C2, and then C2 times C1. Okay. Any questions on that so far? We're doing great. We've multiplied all this stuff out. Now we just gotta throw it all together to get our expected genotype frequencies for that S1 generation. For the C1, it's pretty easy. We just look to where the C1 is and we'll say, okay, it's 0.216. So the expected genotype frequency for the red-eyed flies is going to be 0.216. Then let's go over to the C2. That's pretty easy too. There's just one C2 number. So that's the expected frequency for the white-eyed flies. So we're expecting about 21.6% of the population to have red eyes and about 28.6% of the population to be white-eyed. Then to get the rest, again, this should also add up to 100. So this is another tip for, this should also add up to 1, but the percentages would add up to 100. So the rest is going to be the heterozygotes. So instead of just doing one of them like we did for the homozygotes, you're gonna add 0.249 to 0.249, and then you'll get how many you expect to be pink-eyed flies. So that's our expected one. Then you'll do this whole Punnett squared dealio. Not the Punnett square. You'll do this Punnett square dealio for calculating the expected frequencies. Then you'll do this genotype frequency, which is the super easy number we saw divided by total number for what we actually observed in the S1 and the F1 generation. You'll get these frequencies like we had gotten above, and you'll compare to what we expect to see. If the difference is greater than 0.05, then you'll say that they don't match, that they're pretty different. And we're not going to be doing statistics like we were in this lab. It's just saying, like, say the expected frequency of the pink-eyed flies was 0.49, and we actually only saw 0.2. Then we would say they don't match. Something is going on here. It's not meeting one of the requirements for Hardy-Weinberg equilibrium.
Yeah.
Someone isn't muted. Okay, so does anybody have any specific questions? Or if not, I will jump into kind of explaining this in more detail with the whiteboard.
Yeah, I have a question. So for the expected frequencies for S1, what exactly did you do? Like for C1, how did you get 0.216?
So the 0.216 for the expected frequency is the allele frequency of C1. Whoa, sorry, getting ahead of myself. The allele frequency of C1 times the allele frequency of C1. So it's the allele frequency of C1 squared, basically. So
Okay.
in this box, you're just going to be multiplying C1 times C1, and that'll get you this. And then
Multiply by 100 to get the percent, because the answer is coming like 0.0216.
oh, it's
So
just 0.216.
Oh. Okay. I mean,
Yeah.
I calculated and it came out to be 0.021. So I was confused.
Let me. Is it coming out? Is the math wrong? No. No, so 0.465 times 0.465 is 0.216. Sorry,
What am I doing? Yes. Wait, wow. I mean, that's a glitch in that case, because, okay. I don't know why it's showing this. Yeah, you're right.
no, I had to do,
I didn't even
I don't
have
know, yeah.
my COVID second vaccine. What was showing?
Okay, yeah, it's like
Oh,
105
God.
degrees in my apartment right now, which is why I also am needing to, I'm checking everything that I'm doing on the calculator also because, you know, it's been a crazy week as we've just talked about. So yeah, I believe these calculations are correct, but yeah, at any point, definitely double check. Calculators are our best friends. So, okay, so any other questions about this before I go into kind of another explanation where I attempt to draw with my mouse some not very pretty pictures? All right. Well, if that all made sense to you, if this is old hat, if you understand it completely, you can feel free to go enjoy your evening. Otherwise, stick around and I will do a very fun whiteboard situation where we'll go over the fly stuff again. Okay. Here I go. Can you see the whiteboard? Great. Okay, so to start, as an explanation, like we talked about, we have these flies in our jar, million little guys hanging out in the jar, mating, having babies, etc. So this is the S0. Then we take one little fly. Sorry, oh, that looks like a bunch of flies. This is just one fly. And she's pregnant, and we take her into her own jar by herself. And then she has a few babies, and that's the F1 generation. Meanwhile, over here, there's a bunch of flies all getting it on, like I said, and then they have a bunch of babies that turns into the S1 generation. So there's a bunch of flies in here too. So that's the premise of the experiment. Now, when we first started with our S0 generation, we came in and we counted how many flies there were with each eye color. So we have our S0. We're just getting started on our experiment. We're taking out all the flies and we're counting them by eye color. And so we get 85 red, we get 29 pink, and we get white, 100 white. So now we want to understand what are the genotype frequencies of this original population. So to get that, we're going to add these up. And I think the math is right on this. 214. So there were 214 flies total in this S0 generation. So to get the genotype frequencies, which is, the combination of the C1, 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 C2, et cetera, that leads to the eye color, we're going to take the number from each eye color, so 85 for red, divide by the total number of flies, and that'll give us the frequency. So for red eyes, we've got 0.397. For pink eyes, we've got 29 divided by 214. So 0.136. And for white, it's 100 divided by 214 equals 0.467. So these are the genotype frequencies in our S0 population. Great. Step 1 complete. This was step 1. Step 2. Now we want to know the allele frequencies. So this is gene frequencies. Oh, that looks like gluten-free. And this is the allele frequencies, AF. And to get that, remember, so we're going to take, so let's say we want the C1. Remember, there's just the 2 alleles. So C1 and C2. So C1 frequency. How many C1s are there in the entire S0 population? So this is a little trick to get that. You're going to take, this one is all C1s. Every single fly that has red eyes has 2 C1s. So you can just take this whole thing and you're going to get 0.397 plus, and remember the pink-eyed guys, they have half of their alleles are from the C1 because they have pink eyes, they have 1 red and 1 white allele. So to get the total number of C1, we have the ones with just C1 plus 1/2 of the ones with pink eyes. To get the white, the C2, the one that codes for white eyes, to get the allele frequency for that, we're going to take 0.467 and also add half of the heterozygotes. Okay, and so, wow, can you move this over here? So with that, you're going to get the total C1 frequency is 465, and the total C2 frequency is 0.535. So pretty even, decently even. Okay, now we have our allele frequencies. We can project out into the future and determine what do we think our future population will look like. That is step 3. Bear with me as I try to erase everything that I've just written. Oh, wow. This will take a while. In the meantime, I will tell a joke. What is the worst vegetable to have on a boat? You can type in the chat if you think you know the answer. Not seeing any answers. All right, the worst vegetable to have on a boat is leaks. That one gets me every time. Okay. So step 3, now that we've erased all that, we have our allele frequencies, and we're going to try and determine what we think under Hardy-Weinberg equilibrium the population genotype frequencies will be for the next generation. So you can do this different ways. I was taught to do this with this p squared plus 2pq plus q squared. And I don't know if that's how it was introduced to you. You could pretend this is C1, C2, C1, C2. So you can do it this way if you like. If you're more equation minded, you can do that. But I like this square. I think it's a nice way to do it visually. So we'll go through this. So you're gonna basically set this up, do this little thing, and then you'll have your 0.465 for the C1 frequency, 0.535 for the C2, 0.465 for the C1 on the top, 0.535 for the C2 on the top. And what you're going to do is just fill out the square. There's going to be 4 spots, so you got to fill them out, and it's just going to be multiplying the 2 sides. So we've got 0.465 times 0.465, and we fact checked this, and it's 0.216. And then we've got, so you've got 1 C1, C1, and then moving over to the right, we've got C1 times C2, and that is going to be, check my things, 0.249. And then you've got C2 and C1, so that's also going to be 0.249. So these guys are matching. And then we have finally C2 times C2, and that's going to be 0.286. 0.535 times 0.535. Great, we have filled out the square. Now just to get to that final little push, we're gonna be like, okay, let's look for the C1, C1. All right, so that's gonna be 0.216. And then, oops. Where's the eraser? And then we'll look for the C2, C2, and that's gonna be 0.286. C2, C2, and then, so how many heterozygotes will there be? This is the C1, C1 homozygotes, so the red eyes, and this is the C2, C2 guys, so the white eyes, and then to get to the heterozygotes, you have to add these 2 together. So just remember that the C1, the homozygote C1, C1 is pretty easy. It's just right there. It's the 0.216. The homozygote C2, C2 is also pretty easy. It's right there, just the 0.286. But to get the heterozygotes, you have to add those 2 other squares together. So when you have the frequencies, remember that you're supposed to use all 4 squares. The homozygotes only use 1 square each, but the heterozygotes, you're going to add the 2 squares together, these diagonal squares. So now you have your expected gene frequencies, and I'll write them out after I stop erasing. Let me try and think of another joke. My grandpa's favorite joke was this grasshopper walks into a bar and the bartender says, "We have a drink named after you." And the grasshopper says, "You have a drink named Sheldon?" It's actually a really grandpa style joke, I think. Okay, so we have the expected frequencies are 0.216 for the C1, C1, 0.498 for the C1, C2. Sorry, it's getting messier. and then 0.286 for the C2, C2. And remember, these are what we'd expect to see in S1 if we had Hardy-Weinberg equilibrium. So let's see what we actually got. So we'll pretend that we come back into the lab after a few weeks, days, hours, I don't know, however long it takes for these flies to have another generation. And we're going to calculate the genotype frequency. So we count all the flies out, and then we'll get the genotype frequency. So if you look at the table, now we have now we'll add what we actually saw in the S1 generation. So what we saw was 83 red-eyed flies, 32 pink-eyed flies, and 106 white-eyed flies. So let me add those up on a calculator because calculators are great. And we have 221 flies total in the S1 generation. So we're going to go back to that handy dandy step 1, the gluten-free genotype frequency one, and see what the genotype frequencies are for our S1 generation. Now these are the real ones, not what we expect to see, but what we actually saw. So this is going to be 83 divided by 221. That's the red-eyed flies. Then for the pink flies, it's going to be 32 over 221. And for the white flies, it's going to be 106 over 221. So I'll just do these really quickly. 83 divided by 221 equals 0.376. 32 divided by 221 equals 0.11145, and 106 divided by 221 equals 0.479. Okay, so just based on this, remember, these are our expected frequencies, if you can still read my handwriting. Do these actual frequencies look like what we expected? So we expected 0.216 and we got 0.376. So does that within 0.05 of each other? Chat? No. Yes. So they look actually pretty different from each other. So it looks like something else is going on here. Then for the F1 generation, so from this other side over here, you are going to do the same genotype frequencies calculation and compare what you'd expect to what we actually saw, and you're going to see if they're different. And if they're different, then I'm going to stop sharing here for a second. If they're different, then you can say in your post lab, okay, these are different and something else is going on here. Maybe natural selection, selective mating, something is happening that is not allowing us to meet the requirements for Hardy-Weinberg and produce the expected genotype frequencies. So I hope that was a little bit more helpful to kind of go through that process again. That's really similar to what you'll be doing, basically identical to what you'll be doing in your post lab, but I know this is a challenging thing. So does anybody have any questions or want me to go over anything again? But if not, that was kind of a brief, but hopefully more thorough explanation of this genotype allele frequencies.
I have a question. So why did we even go through the allele frequency? I mean, for the second part, we were just checking the gene frequency and comparing it with the gene frequency of the S0 generation or SO generation. But why did we take the allele frequency? Like,
Yeah, that's a good question. So we need to take the allele frequency because to compare the expected S1 to the actually observed S1 frequency, we're not comparing it to the original gene frequency. We're comparing it to the expected, which we use the allele frequencies to calculate. But yeah, that's a really good point. So that's an important thing. We're not just comparing it to what we saw in S0, we're comparing it to what we expect in S1 given the frequencies in S0.
Okay, thank you.
Yeah, good question. Anyone else? All right, well, I think I'm going to cut the recording now because we went through it, but I'll stay on for a few more minutes for questions. Thanks to those of you who are here, it's great. Good to have some little more interaction here, and hopefully this is helpful for those of you watching at home as well. So have a great rest of your week.